Okay, well, let me just start by saying how pleased I am to be here today. And I want to especially thank Elliot and Alta for the invitation. Um, I first want to start out by telling you who did this work. This is primarily the work of Danny Buchheister, my PhD student currently, but it builds on work that I won't have too much time to talk about today from earlier generations of PhD students, including Muammar Mansour, Becky Macaulay-Wrench, and Dan Jones. So, I don't think I need to go into too much detail for this group, but why study how pyrite forms? Of most interest for us today, iron sulfide minerals figure prominently in ideas about the origin of life and early evolution. And so that's really our focus. But I do want to point out that pyrite production and burial control atmospheric O2. So those are very significant feedback between this process and the Earth system state in general. Um, and in addition, sedimentary pyrite is a really important archive of environmental proxies that we use to understand past environments. Um, and last but not least, there's just a basic curiosity about how pyrite forms in nature. And even for modern sediments, the processes involved are still being debated. So in this talk, we're going to focus on the fact that iron sulfide minerals figure prominently in ideas about the origin of life. And a significant proponent of this line of discussion is Günter Rechtershoyser, who in the late 80s and early 90s proposed that pyrite was at the center of the evolution of some of the first metabolic cycles. This became known as the iron sulfur world hypothesis. And the reaction of interest is shown here at the bottom of this slide. And you can see that the delta G under standard conditions is favorable. It's also favorable in the environments that I'm going to talk about. So thermodynamically speaking, at least, this is the metabolism that is on the menu, and it's a missing metabolism, meaning we've never met a microbe that is known to conserve energy by carrying this out. So that's also a tantalizing sort of aspect of this work, is that we're hunting for this missing metabolism that may be related to early metabolism. So if you're interested in this topic, I want to point you toward a very recent review, spearheaded by Muammar Mansour, but with a cast of brilliant people. And this review concluded that one of the interesting conclusions is that although we widely accept that microbes are important in the production of pyrite, most attempts to produce pyrite in the presence of microbes in the lab have failed. And so for some reason, it's hard to get microbes to perform this trick in the lab, and we don't really understand why. Luckily for us, we've got a great model system to give this a go. And so I'm going to introduce you to the Frasassi cave system. It's in central Italy. This is a plan view map of the system, and you can see these splotches of blue. And those splotches of blue are subterranean lakes and springs that bring sulfidic aquifer in contact with the cave atmosphere. So essentially, these are points of access to a deep subsurface aquifer that we can actually access this ecosystem, which is about a half a kilometer beneath the ground surface. Okay, and most exciting for us today is that the water-filled passages are choked with this stuff. This stuff is we call them hanging gardens. They're hanging biofilms, and they're in contact. The bulk water around them is anoxic and sulfidic. It has a proterozoic-like chemistry. It has a lower sulfate concentration than the current modern oceans. So there are many aspects of the chemistry of this aquifer that smell like Proterozoic. But most exciting is that these biofilms are really concentrated with pyrite grains. This is what it's like to work there. This is actually Becky Macaulay-Wrench in the teal suit, and Dan Jones is in the red suit in the middle. So this is a historical archive photo, but this is kind of what it's like to work in this environment. I'm going to try for this video. Let's see. I hope you can all see that. This is a diver footage showing these hanging gardens. You can see that the water is clear at first, and then as the diver moves through, you get significant silting. So it's a really challenging place for divers to work. And this is just some footage that shows you, nope, this is that same one. Hang on. Pause. How do I go forward? Interesting. Here we go. This is going to show sampling the biofilm material. And so we depend very heavily on relationships with highly technical science divers to do this work, and they are crucial to our operation. And I just want to give a shout out to them. They are co-authors on our papers, and their intellectual contributions are crucial. So when you bring these things up to the surface, they have a lot of ultrastructure. They're super interesting from a bunch of points of view. Here's a nice view of one that looks like it has a churro dessert-shaped format. If you squint, you can see some growth rings on the fins. So they're very interesting. They have high diversity, which is a real pain, and there's lots of dark matter. When you look at them using scanning electron microscopy, you can see the density of pyrite framboids and also euhedral grains. So in this image, you can pick out both framboids, which are composed of smaller euhedral grains of a variety of particle sizes. And then you see a lot of amorphous material. And that amorphous material is organic carbon rich and also contains significant silicate clays. So we wanted to try to find this missing metabolism. And so we designed an enrichment medium that we inoculated with that biofilm material from the hanging gardens. The reaction that we want to happen is the Vector-Schweizer reaction, shown there at the top in bold. We want FeS, or McKinowite, to combine with H2S and produce pyrite and hydrogen gas. So very simple setup. We made FeS, and we designed this medium to enrich that missing metabolism. And I have to tell you that after a year, we weren't able to detect anything in the headspace of these enrichments, and they were almost discarded. And I said, no, don't throw that out. You need to look at it at least once under the microscope. And luckily we did that. And it turns out that they're full of cells. And the cells appear to be associated pretty closely with the amorphous carbon-rich material and with the mineral grains, which are opaque in this transmitted light micrograph. So very exciting. The abiotic controls are beautiful in this experiment. They just look like amorphous McKinowite. On the right, you can see how it looks under light microscopy, whereas the enrichments seem to have these opaque mineral grains that are associated with the biofilm material. And so this is exactly what we were hoping for, but the question is, what are those mineral grains and what is really going on? So when you look using scanning electron microscopy at the enrichments, these are about 1 year old, you see these clusters of minerals that are embedded in this sort of broader matrix of more fine-grained material that ostensibly is the McKinowite. And the question is, what are these pseudo-framboids? You can see that they don't really look like framboids beyond their spherical shape. And so that's a bit of a mystery. But at this point, we need to know what they are. So here's a close-up of them. You can see that they're aggregates of sort of blade-shaped grains. And so the next thing that we did to investigate their composition was EDS, which is energy dispersive X-ray spectrometry. In this beautiful scanning electron micrograph collected by Danny, please ignore the glamorous microbial filamentous cell in the middle of the image and focus on the mineral aggregates. And you can see on the right that they are very rich compared to background in both iron and sulfur. So that would suggest that they're made of an iron sulfur mineral. And in fact, the enrichment cultures, not after 1 year, but after 2 years, they show visible signs of clearing. And so on the bottom, you can see the control bottles that have been incubated for 2 years, and they're black as night because of the fine-grained, nanoparticulate machinowite that we made as a feedstock for this medium. But the enrichment cultures are cleared. You can see material in them, but they're substantially cleared, which suggests that there is mineral growth and or aggregation of those minerals over time. So back to EDS, this is another image of the enrichment mineral particles and showing both iron and S concentrations. And so this is a pretty nice example of how we can find out more about these mineral grains. EDS can be made semi-quantitative if you have a lot of data points and if you have the right kind of machine. And so we did a lot of measurements of these mineral particles using EDS to try to get a sense of the ratio of iron to sulfur in these particles. And this is the results of those experiments, an example of a scan. So if we were looking at pyrite, we would expect an iron sulfur ratio of 1/2. Whereas if we were looking at the original machinowite, which is FES, we would have a ratio of 1. And so in this scan, you can see that the ratio is closer to 1/2. It's actually very close to 1/2. So that suggests that this material is FES2. So after many, many scans, these are the data. And let me talk you through this. The control FES on the far left. So we're looking at the ratio of iron to sulfur. And remember, what we're expecting for different minerals is shown on the far right of the slide. So in the plot, the leftmost box is the control FES. That's the machinowite that we made. If you just age that in an abiotic control, for some reason, the excess iron goes down and you get a ratio around 1, which is still what we expect for machinowite. In contrast, if you look at the enrichment iron to sulfur ratio and the biofilm original inoculum ratio of iron to sulfur, you get a ratio around 1/2, which is what we expect for FES2. And just to zoom in on that side of the plot a little bit, here you can really see that there's excess iron, a little bit of excess iron potentially, but this is probably within the limits of the quantitation of this approach. So essentially what we expected to see is what we saw. So the enrichment and the original biofilm inoculum contain FeS2, and that's to be contrasted with the machinowite that we added into the medium. So can we determine the mineral phase of these materials? And the answer is yes, we can try that using Raman spectroscopy. Raman spectroscopy is a non-destructive spectroscopy that you can use on aqueous samples, and it's sensitive to the bonding environment of the molecules that it interacts with. And so this is a squiggle and match, a kind of approach like many other approaches you may have encountered. And on the top, you're seeing different colored lines represent different scans of one of the enrichments. So in a variety of spots, the laser moves around and you get scattering at different wave numbers. And the intensity of that scattering at different wave numbers makes up the curve that you see. And so in this LV23-1b enrichment, which is a first generation enrichment in this experiment, you can see peaks that are pretty nice. There are two references, pyrite references here on the slide. One of them is in the lower plot. That is reference pyrite. And there's also 3 lines that represent data collected in a different study for reference pyrite. And what I want you to notice is that it looks like a pretty good match, but there is some variation between the reference pyrites. And so we expect some variation in these pyrite spectra, but it looks pretty good. If you were to ask, well, is it maybe marcasite? Is there marcasite in there? Marcasite is another phase of FES2. And the answer is no, it doesn't look like marcasite at all. Is it McKinowite? Here are the vertical lines you can see are the lines that you would expect for aged McKinowite. And so if you compare those lines to the enrichment, they don't match at all. Not a good match. However, if you look at the abiotic control, which should be aged McKinowite, you do get a good match with that reference spectrum. So it does, it all fits together in the sense that the abiotic control has the aged McKinowite that we put in, and it matches the Raman spectrum for that. So I did show you a spectrum that didn't seem to contain marcasite. However, some of the other enrichments do seem to contain both pyrite and marcasite. And these are spectra showing the matches between those 2 minerals and the scans of the enrichment. And this is interesting to us because it matches data that were collected by Momar Mansour a decade ago using optical microscopy techniques, and he did detect both pyrite and marcasite in the original biofilm inoculum material. So this is not entirely unexpected, but it is very curious, and we don't understand all of the reasons why marcasite is present. It shouldn't be present. It's not supposed to be stable at this pH, which is near neutral. Okay, so what's the taxonomy in these enrichment cultures? There are 2 enrichments shown here in the top 2 bars and the original inoculum shown in the bottom line. And this is too much to take in at the speed of this talk, but I'll draw your eye to a couple of things. There does seem to be an enrichment in certain taxa compared to the inoculum, and those enrichments are primarily Desulfobacterota, which used to be called Delta proteobacteria, and Campylobacterota in pink, or sorry, in the dark blue color, dark teal blue color. And that used to be called Epsilon proteobacteria, who are, if you're as old as I am, you can remember these things. So Campylobacterota and Desulfobacterota are enriched, and there are taxa that have disappeared, like planktomycetes have disappeared, alpha, beta, gamma proteobacteria have disappeared almost entirely. So the community is changing, which is comforting because that's what we set out to do in an enrichment culture. We also find that the enrichment communities are significantly lower in diversity, taxonomic diversity, than the original inoculum. So the original inoculum is shown in the blue squares, and 2 different enrichments are shown in the gray triangles. And so this is exactly what we want for an enrichment culture. We want a signal from the original enrichment. We want certain taxa to be enriched and others to disappear. And we want the overall diversity to go down, and that's what's happening. Okay. So let's see. I have 3 more minutes. Is that right? I think so. So the emerging conclusions from this experiment is that the enrichments are producing FES2, including pyrite primarily, but also sometimes marcasite. And they're producing that from FES, from McKinowite. And this enrichment community is dominated by Desulfobacterota and Campylobacterota. And if you know those phyla, you know that their claims to fame are sulfur cycling. So this is consistent with a sulfur cycling community being involved in whatever transformations of the minerals are going on. So I could tell you a whole lot more about the system, and I'm tempted to do that, but I'm going to stop and acknowledge some people first and leave lots of time for questions. And I have other slides that I could show you if the need arises. So let me thank most of the graphics on this slide are caving organizations that have volunteered their time. Cavers are some of the most incredible citizen scientists I've ever met. They're absolutely fantastic, and they rarely want to be paid for their time. Um, some of this work was supported by a Hansa Fellowship and by the Joint Genome Institute. The beginnings of this work were funded by the NASA Astrobiology Institute, if you remember what that was, and also by NSF. And with that, I will take questions.